Hello, my name is Laura Modelli and welcome to Science for Education, supporting teaching and learning in science education. In this episode, we are going to learn a little bit more about DNA by doing an activity, strawberry DNA extraction. In our last video about DNA, we talked about how every living thing is made of cells and every single cell contains DNA or deoxyribonucleic acid, which is a material that carries genetic information. In strawberries, this genetic information determines the physical characteristic of the strawberry, such as the color of the strawberry, the size, the sweetness, the shelf life, etc. I chose strawberries for our experiment because each strawberry cell contains 8 chromosomes, which is more than what many other fruit cells have. Scientists use the word octopoid to describe strawberry cells. Octo is the Latin prefix meaning 8. In this activity, we will need a couple of strawberries, some salt, some water, ice-cold rubbing alcohol, liquid dish soap, small cups, and here I am using beakers, a heavy-duty zipper storage bag, and some tools like scissors, a quarter teaspoon, a tablespoon, a stirrer, and a toothpick. First, you must place the rubbing alcohol in the freezer for at least 30 minutes. Keep it cold until you can use it. Next, blend 1 tablespoon of water with a quarter teaspoon of soap in a cup. Add a pinch of salt. The soap in this mixture will open the cell, while the salt helps untangle the DNA. Next, we are going to put the strawberry in the storage bag and mash it until there are no large chunks. Then you will pour the soap mixture into the mashed strawberry. Seal the bag and use your hands to mix everything together for 1 minute. What happens here is that the soap breaks down the fatty oils. Think of it as when you wash dishes using soap. The thin membranes surrounding strawberry cells contain fatty molecules called lipids. The soapy solution dissolves the lipids, which exposes the inside of the cell's nuclei. Inside of the nuclei is the DNA. What is the function of the salt or sodium chloride? It breaks the protein chains in the nuclei and allows the DNA to separate from the protein. Next, we are going to filter the strawberry. Rub the filter over the top of our second small cup or beaker and secure it with a rubber band. Make a small dent inside the filter to collect the goop from the bag. Snip a corner of the bag and pour your mixture into the filter. Let the mixture drain through the filter for at least 10 minutes. You will have liquid in the bottom of your cup. If there's not a lot of liquid, then that's okay. Assemble your test tube. Get the alcohol out of the freezer. The colder the alcohol is, the more DNA you are likely to see. Make sure you do this next part with an open window nearby because alcohol vapors can be harmful. Measure and pour about 25 milliliters of rubbing alcohol into your test tube. And then carefully pour in your strawberry mixture. Put a cap on your test tube and wait 5 to 10 minutes. DNA dissolves in the water and it will be invisible. Therefore, to be able to make it visible to the eye, we use the cold alcohol to cause it to undissolve or precipitate. See where the layer of filtered strawberry touches the layer of alcohol? Look at the very thin white strands forming there. That is the strawberry's DNA. The DNA strands float above the layer of water which is denser and heavier than alcohol. It may look like feathers, small balls, or wispy clouds, Wait for another 5 to 10 minutes before we can see the DNA float to the top. Collect the DNA by carefully gathering the DNA strands on your toothpick. I hope you enjoyed our lesson today about how to extract strawberry DNA. And if you feel inclined to learn about different concepts of science and want to keep up to date with our lesson videos that come out every Monday, you may hit the subscribe button below. And I will see you next time.